and welcome, Felipe, and we look forward to hearing from you. Thank you so very much.
Well, thank you, Olga, so much for this introduction, and I'm going to share my slides. Hopefully you see them now. And slideshow. All right. So it's really a pleasure to be back. I think I've done this last year, and hopefully we'll have an opportunity to do it in person again when all this is over. But I will talk about our FragPipe computational pipeline, and I'm really excited about this. I joined Twitter so I can tweet about it. As you know, I do it with some regular frequency because I'm really excited about what we are doing and how this pipeline is working now and what we have for it in the future as well. I'll talk about quantification and FragPipe, but also give you a brief overview of the whole sort of functionality of FragPipe, including post-translational modification analysis, because this is where sort of where we started with developing this pipeline. About my lab a little bit. So we are a computational lab, but I'm also directing a proteomics facility here at the University of Michigan. So we try to couple computation as well as data generation, and we even moving towards sort of more wet lab proteomics as well. I want to use this very old slide. This is a pre-pandemic slides. It's actually a view from my window. Well, it's a table outside of my window. We don't have snow now, and we're not wearing masks again, so that's nice. But anyways, I should update this slide. I really want to acknowledge my lab. Again, this outdated slide. The current sort of FragPipe team, core of the FragPipe team are listed here, especially Finchao, Felipe, Guozi, but also Danny, Dan Palaski, Kevin Young, Kai Lee just recently joined the group, Jeannie and Leo more on the proteogenomics side. But also want to really acknowledge my former lab members who contributed significantly to FragPipe at various stages. FragPipe is extremely easy to use. Hopefully some of you already have experience with that. Those who don't, I hope I'll convince you to try it after my presentation. But we have a graphical user interface, GUI, which is a screenshot of that, kind of an older version of that we see on the screen, where we have this concept of workflows, where it's extremely easy to use for you as a user because you could just select one particular workflow that you like. That's already predefined for you. For example, a DIA-based spectral library DIA analysis, or SciLac with TMT, 10, and load, and all settings are going to be already loaded in FragPipe for you. All you just need is select files, load the files, and click run. That's why it's so popular. Of course, you can customize workflows, you can save a workflow as your custom workflow, even share a mail to someone. People often contact me and I customize the workflows and email back and do things like this. So that's why it's very powerful. But in addition to that, we also have command line versions of this workflow. And in fact, in FragPipe 18, which we'll be releasing before the SMS, probably next week, we'll have a first release of what we call headless FragPipe, where you can do everything that you can do in the graphical user interface, but through the command line. That's very nice. And we have other ways of running this pipeline as well, including what we call philosopher pipeline. So you can run it on the cloud, you can run it on desktop, on Linux, HPC. That's why it's a very powerful system, because it's platform independent, Windows and Linux, and it's scalable to very large amounts of data. So I'll talk about some advantages of FragPipe, of course, with a focus on quantification workflows that we have here. But I'll go through this post-translational verification analysis first a little bit as well, because this is where a lot of strength of our pipeline, a lot of strengths are in this sort of comprehensive PTM profiling as well. So let's talk about that part first before we move to the quantification. And I can already mention that why we started developing this pipeline in a way in the first place is that we this goes back to my first project in the mass spec when I just joined the Rudy Abersold lab, even before Olga became a sort of office mate. My very first project before I was officially working there was to try to understand what are all those high-quality spectra that we identify, we collect, that we have no identification. Thinking that they would be a spectra corresponding to a post-translationally modified peptides, or peptides harboring a mutation, or maybe novel peptides. And of course, a lot of spectra were also chemical artifacts. And, it's really, now 20 years later, or how many years? 20 years later now, we have all this suite of tools, but in fact, it's because we really wanted to go deeper in the data and identify all these peptides. So this dark matter of mass spectrometry-based proteomics is why we started developing as a sort of our last iteration of our attempts to try to solve this, to develop a new search engine that could allow us to identify those peptides in a much more efficient way, comprehensive way, and also much faster way. So that's how we end up with MS-Fragger search engine that uses ultra-fast searching, implements ultra-fast searching based on our fragment ion indexing strategy that we sort of discovered and developed over the last 5-7 years. And indexing is a technology that allows us to index the data, whether it's this protein database predicted via the ions for all peptides, or we can index experimental data as well. So it's a technology that allows us to index the data and then search that data really, really fast. That's why MS-Fragger is 100 times faster than a conventional search engine. So based on that, we started, based on this advanced and on MS-Fragger search engine, we realized that we have to put other pieces in place to really create a more complete pipeline that people can use. And we developed Philosopher toolkit, and Felipe developed most of it, where we basically implemented our transproteomic pipeline components, the key components, peptide profit, protein profit, and more recently, iProfit, PTM profit, as well as various scoring modules, FDR scoring modules with all various options, razor approach, peaked FDR, sequential filters, and so on and so forth, as well as reporting and generation tools. So that's a Philosopher is a toolbox that's a key part of FragPipe as well, that sort of runs the transproteomic pipeline components, as well as FDR filtering and reporting tools. So with respect to PTMs, before we jump to the quantification, because it is one of the big parts of our success is this open massive set search strategy that we developed. And that's an alternative strategy for identifying post-translational modifications that goes beyond what has been tried before, or still tried in other tools, which is iterative or multipass searches, sequence tagging, or a dependent peptide approach that's probably best known through its implementation in MaxQuant. So open massive set database search strategy is a highly efficient strategy that's also very, very simple conceptually in a way. And that's why we really like that. So it's illustrated on the slide that you see here. So a conventional closed search that we're all familiar with is where you specify a very narrow tolerance of maybe 20 parts per million, for example, for a modern instrument, and perhaps allow isotope errors and most common modifications such as methane oxidation and perhaps internal acetylation. But other than that, you won't be looking for those peptides. You're not looking for any other modifications or unexpected mass shifts. And that's a conventional closed search, right? So you're really looking at like, this 20 ppm around the expected mass of the peptide. Now, the open search is the sort of the opposite extreme of that, where you can have no restrictions at all. So you can search for a peptide in the database with any mass shift with respect to the measured experimental mass for that MSMS spectrum, right? So you allow any possible mass shift. So it's a complete open search. In practice, we typically do from minus 150 Dalton to 500 Dalton, or you can go to 1,000 Dalton, but in principle, it could be a completely unconstrained. And then we have this really nice strategy that we call massive set search, where it's a combination, it's something in between the two. The closed search and open search is where we search with a narrow tolerance of 20 ppm, but around multiple notches. For example, we can allow unmodified peptides with a mass shift of 0, but also we can specify we allow acetylation, say phosphorylation, and we'll search with a plus minus 20 ppm around mass shifts that correspond to those modifications, right? And this could be a really long list. So our common mass offset workflow in FragPipe, for example, has about a few hundred, maybe 300 most common post-translational modifications. And it's a really good alternative to full open search. And our glyco workflow and lab modification workflow, that's now, it's a really huge field for my lab. Those areas are based on this concept of a mass offset search. So using full open search, we can search for any mass shift, and if you run it on any data, standard data, you can use it as a quality control. I do it on every data set that I'm running. I'm running a ton of data sets. I'm like a beta tester, and I should be doing a lot of other stuff, but my favorite activity is to run FragPipe, frankly. So anyways, I run a lot of FragPipe, and I do a lot of QC analysis, and you just, even if it's a regular search triptych digest, do it, because you'll find a lot of artifacts, a lot of unexpected modifications that you can optimize your protocols and change your search parameters based on that. So this is how a histogram of mass shifts would look like for a typical open search on a regular digest. You'll see a ton of those mass shifts corresponding to common modifications, and the negative mass shifts corresponding to neutral losses or semi-triptych peptides. If you don't allow them, you'll find them through open search as well. We typically would get, a very good booster number of PSMs, 30% or more identified. So as I said, you can do it even if nothing else. You can just do it for sample QC. But we have a huge user base now, hundreds, thousands of people are using that for other things, chemical proteomics. Probably all major pharma companies now license our tools, because they do covalent modifications, they do all sorts of PTM and electrophile screening libraries and stuff. That's really great for that. But RNA cross-linking, all of that can be captured through this common mass offset search strategy or open source strategy. We extended a basic open source strategy more recently to introduce what we called localization aware open search and shifted ions. I do not have time to go into discussing this extensional algorithms of Inchao. Let this work with the help of Guozi and others. So it's published in a year and a half, 2 years ago now. But we really extend open search to also capture shifted ions. So in conventional open search, you're basically identifying a peptide from unshifted YNB ions that don't have the modification. And it's actually sufficient to identify most of the peptides, even though you lose half of the information, up to half of the information. But in the localization aware open search, it's now part of MS-Fragger is a localization aware open search mode, and fully supported on FragPipe by default, actually. We also have this modified fragment ion indexing strategy using what we call shifted ion indexing. We can capture peptides with fragments that contain modification as part of the open source strategy. That really gives us a nice boost in the number of modified peptides we can identify through open search. But also allows us to localize the mass shift on the peptide, which was one of the limitations of our original open source strategy. So if you apply something like open searching to interactome data, for example, where we have bait proteins that are enriched using affinity purification, so they're very abundant. So these are not typical samples. These are samples where one protein is highly enriched in a sample. So if you look at how many PSMs we have for bait proteins in this affinity purification mass spec experiments, and here we are reanalyzing the data from the Bioplex study, from Bioplex 1 from Steve Gige's lab and Wade Harper's lab. So for Bioplex 1, we reprocessed 64 million spectra. It took us only a few days. With MS-Fragger, it would take you years probably with sequest. So what's interesting is we look at how many PSMs we have per bait protein that we identified in open search compared to conventional clock search, like they did in the paper. or like most people would do still. I mean, that's amazing. We get, for some proteins, we get, 100 times as many PSM. So here's an example of one protein, ETH-E1, where we go from 48 peptide ions found in a conventional closed search to to over 2,000, almost 2,500 found in open search, right? A huge boost. So this is, there are 1,000 dots here. There are 1,000 APMS runs, 1,000 proteins used as bait proteins. So for each one, we show the number of PSMs in open search versus closed search. And you see on average, we get 400% increase. So 4 times as many PSMs. But for some of them, like this one that I show, you have 50 times as many. And interestingly, a lot of proteins that show the highest number of the highest boost, the number of peptides that we identify in open versus closed search in this data. If you do a gene enrichment analysis, you'll see that those proteins are enriched for a category, genetology category called small molecule metabolic processes, which suggests that this is the proteins that modify other proteins, including themselves. So perhaps this is why they're so heavily positionally modified in this data set. And that's why we get the highest gain in number of PSMs we have when we go to open search, capturing all those modifications compared to the closed search, looking only for no modifications or only oxidized methionine. So in the last few years, we worked really hard, and again, we have a really great team developing MS-Fragger and all other tools in the pipeline. So in addition to the localization aware open search that we developed, we have mass calibration, we have search parameter optimization, we have de-isotoping, we have support for FAMES, as well as timsTOF ion mobility data, we have monoisotope correction, we have improved scoring now, improved signal processing, neutral loss removal. I mean, I have a ton of work that went into MS-Fragger, as well as in all other tools. So this is really in active development. So a key tool for PTM analysis in FragPipe that we have is PTM Shuffler that Danny Geisler, graduate student in the lab, developed, or published an MCP, what is it, less than a year ago, that if you do PTM analysis, it will summarize the site localization properties for each mass shift, spectral similarity, retention time shifts for each mass shift. It will summarize the abundance of peptides in modified form compared to the unmodified form. So I welcome you to go and check out this paper. You can find and characterize unexpected novel PTMs with MS-Fragger coupled with PTM Shuffler. So it's very useful. We have an example here of finding some interesting mass shifts in, for example, in a synthetic data set from Bernard Kuster lab, where with the help of PTM Shuffler, we were able to distinguish one particular modification from a neutral loss of 18 that's typical in this data to actually been located to a C-terminal lysine, which we think is a cyclization effect, which is a very interesting sort of unexpected byproduct of peptide synthesis that I think people should be aware of. And tools like FragPipe with MS-Fragger and PTM Shuffler allows you to identify this type of events and sort them out and figure out which are neutral losses, in which are modifications that happen prior to mass spec. You can run it again for QC. This is where we run this pipeline as part of our CPTAC efforts. We are a member of CPTAC consortium, where we work with some ridiculous amount of data, TMT data. But here we run it as a QC run using CPTAC 3 harmonization data generated at 3 different sites, Johns Hopkins, Broad Institute, and PNNL, where they use the same samples analyzed through harmonized pipeline, but at different sites. And we clustered here different mass shift PTMs that we find in each site in different plexes, or if you have replicates. So each row here is a different replicate. And you see how the data clusters based on the site where the data was generated. So there are some PTMs or artifacts that are specific to each protocol, even though the protocols are harmonized. But even within the same site, sometimes we see a different sort of PTM profiles. And that's because there is a lot of variability in sample prep. For example, you could have a lot of formulation or other PTMs, maybe due to differences in TMT labeling that happens, on different days, and so on and so forth. So we can really, with our tools, get this picture of all PTMs. And in this case, most of the things we see are probably chemical artifacts, but when you run it in biological, it's sort of, we also recover interesting biological PTMs through similar analysis. So another thing that I will not talk about at all now, except just to give you this advertisement for a huge effort in the lab, is glycoproteomics and labile modifications in general. And we extended MS Fragger with our common our master set workflow, as well as PTM Shepherd. We extended this to glycoproteomics, and it's a super powerful workflow, and based on our benchmarking, it's the best tool for glycoproteomics. And others agree with that. And as part of the HUPO initiative, there is a benchmarking data set where we've done this benchmark, and you'll see the results announced very soon that we've done really, really well with MS Fragger Glyco, as well as a more recent work on estimating full discovery rates for different glycan structures that we published in MCP more recently. So this is the earlier work by Dan, and we have a new one in MCP that further extended this. So we have a glyco workflow in FragPipe that's also fully compatible with label free quantification or TMT quantification of glyco TMT workflow. So if you are into glycoproteomics, I strongly encourage you to explore this, what we have to offer. So now let's move to quantitative workflows in FragPipe, and we'll talk about label-free quantification first, and then TMT. I don't think I'll talk about SciLac, and I'll conclude with some comments on our DIE efforts and what we have now, which is actually really great. So label-free quantification. So we developed a key tool called IonQuant. That's the part of FragPipe. And IonQuant basically is a label-free quantification tool, or rather, it's the MS1 quantification tool. It fully supports SciLac and other labeling workflows as well. But talking about label-free quantification with IonQuant, basically it has three key parts to that. First, we have advanced single processing, so it's fully compatible with TeamStuff passive data, and in fact, probably most of TeamStuff users who are using label-free quantification are using FragPipe with MS Fragger and IonQuant, because it's super fast. It's completely, it's day and night compared to other tools. But also even for thermal data, we have key advantages is that we have highly efficient match between runs implemented in there. So not only optimize single processing, but we also have match between runs with full discovery rate control. And full discovery rate control for match between runs is the third key part of IonQuant. So I'll briefly go through that. So I already mentioned that it's super fast because we index the data like MS Fragger, we index the data. So that's why tracing the peaks and everything is super fast. Including for TeamStuff data where speed is always an issue, but also for FAMES data on thermal instruments. We have FDR control match between runs. And again, we have complete support for labeling-based analysis in IonQuant as well, including SciLac. So on signal processing, I already mentioned that we have optimized peak tracing. I mean, we put a lot of efforts in trying to optimize all of that, including boundary detections, smoothing, which is always a challenge, especially for low abundance signals. And the settings in IonQuant are very, very flexible. So if you're an advanced user, you can really, play with a lot of these settings for how you do signal processing. But again, if you're not so advanced user, you just want to run what we think is optimal. You just load the FragPipe workflow, and it has all settings already that's set for you that we think is the best analysis. So the match between runs, the concept is well established. In match between runs, you basically have a donor run, an acceptor run. The donor run is a run where we identify a peptide from a mesomer spectrum, that we did not identify that peptide in our acceptor run, and you would like to transfer that ID, you would like to find that MS1 peak through retention time, ion mobility, if it's TIMTAF data, alignment from the donor run to the acceptor run. So, and you can have maybe hundreds of runs in a data set. So for each run, you would have maybe potentially hundreds of runs that are donor runs for which you can transfer the ID. And you can either transfer from runs that are more similar to the donor run, to the acceptor run, or you can transfer from any run. So all of these are highly customizable settings in FragPipe. By default, we transfer from all runs, but on very large data sets, you can restrict that to maybe top 10 runs or even less. So, I mean, this is kind of very well established. I mean, the advantage of our algorithms for match between runs here is that we have a very fast and highly efficient alignment between different runs. We use a neighbor-based alignment, and I want to give a shout out to a tool called Metamorphosis. Metamorphosis has this flash LFQ that has very nice. This had a good idea for doing this a neighbor-based alignment, and we implemented that, and it works really great. So
Alexei, there's a question which maybe
yes.
Someone is asking, what do you mean by decoy on this slide?
Oh yeah, so decoys. So these are not decoy identifications like decoy sequences in a database. Here to implement sort of false discovery, false transfer estimation for match between runs, we need to create kind of signals that we try to transfer from the donor run to the acceptor run that actually don't exist. So what we do here, and let me see if it's on the next slide. I don't really have that. But what we do is that we take a real, so in a donor run, so here we have a peak that we identified here from MS spectrum. We have mass to charge ratio, we have retention time of that peak, right? But we need to create like a decoy, a fake signal that we want to try to transfer and see if we by mistake also find that. So we shift mass to charge ratio of the peptide by a certain mass. So so this idea comes from the early work from Dick Smith's lab from probably 20 years ago. Vlad Pityuk worked on that and others. So we referenced this idea in Ion Quant Match Between Run paper. So we basically shift identified peptides by certain mass to charge ratio to create signals that shouldn't exist. So we shift them by like, I think 11 Dalton or 13 Dalton and try some other shifts. So those mass shifts don't correspond to any common PTM. So we basically take an MOZ that shouldn't be in the data. We take identified MOZ, shift say by 11 MOZ or 13, I forgot, and go to the acceptor run and try to find it there. And we shouldn't find it. If we find it, we call it a decoy transfer. So we basically create these decoys, try to transfer them, and then like this peptide profit, so all these things that we develop for target decoy analysis, we basically have scores of for each transferred ion, we calculate the score based on like how close they are in retention time to the expected value for the ion mobility, and like the isotope profile, isotope pattern in the donor run, does it match the one in the acceptor run? We calculate the score, and then we can see like what are the scores we get for decoy values that we transferred, right? So we have like, so the blue is the scores of those decoys, and the red are scores of target ions that we transferred, and we kind of like the model this decoy shape of the decoy distribution, and then we feed it to the target distribution to model this kind of like the negative tail here. So through this analysis, we estimate basically posterior probability of correct transfer, where basically decoys here allows us to estimate the posterior probability, and then the false discovery rate for that of match between run transfer, right? So that's what we do here. So other tools like MaxQuant don't have the false discovery rate estimation, and we do have it, and we control it at 1% ion level false discovery rate for the transfer by default, although you can change that. So that's the sort of the decoy transfer idea here. So we tested this, and I cannot see if there are questions. I cannot see them because I only see my slides. So let me know. Thanks for bringing this up, Olga. So we tested this on a whole bunch of data sets, of course, and like everyone else, we have to compare with other tools. And MaxQuant, frankly, is an easy comparison for us. So anyways, this is comparison with MaxQuant, match between runs, which does not have FDR control. But this is a data set. This is a sort of a popular way of doing things. We have 3 organisms mixed at different proportions in a sample A versus B. So we have A versus B with a human 1-to-1 in both samples, and then we have yeast, and that's 2-to-1, so log-fold change 1 in sample A compared to sample B, and we have E. coli, which is 4-to-1 ratio, mixed at 4-to-1 ratio in sample B compared to A. So it's a negative log2 change of -2. And we quantified more proteins in ion-quant with match between runs and MaxQuant. You see the numbers here. But importantly, as you look at the number of outliers, visually you see that we have much fewer outliers, the tighter distribution of one, of around 1 for human, and less outliers for both yeast and E. coli, compared to, this is the timsTOF data, compared to MaxQuant here, and it's visualized using LFQ bench. So we have excellent quantification performance compared to MaxQuant. On single cell data, for example, this is why most single cell groups now adopted our pipeline, including in Lab from PNNL. So this is his data. We convinced him that our pipeline is great by showing him his result, his data analyzed through our pipeline. So they're our biggest fans now. So this is the Orbitrap Fusion Lumos for replicates with 50 cells with this boosting channel. So you could use, if you do a label frequency, LFQ single cell data, you can use 50. You can do 50 cells, and then transfer. You call them library runs or boosting runs, and through match between runs, you transfer IDs from 50 cells to single cell data. So 50 cells is library runs for replicates, and then you have 11 replicates, so single cell data, but you also have tests for 3 cell data and 10 cell data. And again, we control here with 1% FDR. We don't know what FDR is for match between runs and MaxQuant. It's not controlled. So let's look at the single cell data. Now, obviously, with match between runs, we quantify a lot more proteins and peptides in single cell data. So so here we have 11 dots. No match between run. We have only 250 proteins on average, goes to 750 with match between run, and of course a similar boost for MaxQuant as well. But we have more than in MaxQuant. You see, 0 cell is a good control here. Match between run in MaxQuant kind of like boosted too much the number of IDs in 0 cell data, where because we control it in an ion-quant, we don't have that boost in single cell in 0 cell data. 0 cell is blank data. Ideally, we shouldn't find anything. It's not a perfect control, but still, I'm glad to see that we are not boosting our IDs with match between runs in 0 cell data as much as MaxQuant. Again, it shows that we are controlling the FDR, and we still have overall high number of proteins quantified. So that's the single cell data. And we demonstrated this across a wide range of data sets. Others use this now on timsTOF data on thousands of runs. There is a Cell paper from a consortium, COVID consortium in England, where they run our pipeline. I mean, the pipeline is adopted now, by many labs, and there are a lot of independent benchmarks that people published, sort of, that you can go and look up. Because everything that we show, of course, we're a little bit biased. So we should look at what others get with our pipelines, and you'll be pleased to see that that actually works well. And the speed has always been an advantage for us due to indexing. So again, MaxQuant is probably not the best comparison because it's just so slow. Perhaps they improved it recently, but even against other pipelines, we do really well. So this is the comparison against MaxQuant here, where we have you go from days to hours really when you run this. So more recently, we added a very important module in FragPipe. Before I continue with quantification, I just want to mention that we also added risk scoring based on deep learning called MS Booster, led by Kevin Young in the lab. Work really great. It boosts the number of IDs, and it helps us to boost the number of IDs, especially in cases where the search space is large, where the gray zone is bigger, such as HLA, peptidome data, or non-specific searches of all kind. But it works for any searches, really. Even basic TMT searches. Label searches. So again, a preprint is coming soon, but it's fully functional in FragPipe, and in fact, it's already default for most workflows, called MS Booster. So this is just a slide illustrating that we gain IDs in single cell data sets, but we gain across the board in all data sets, and especially in HLA peptidome data. That's where we have a huge boost, as well as in DIA workflows that we also gain a lot. So let's
Alexei, before you move on, another question related to the comparison with MaxQuant. Dmitry is asking, can you say that the difference was seen in low abundant proteins? And if yes, have you, or is it possible to manually verify what's going on?
Well, the difference with MaxQuant, of course, the low abundance proteins, the proteins that are quantified by a lower number of ions, is where you gain the most. Because if you have more, tools like IonQuant, IonQuant can be run with MSTATs, for example, too, and MSTATs will model peptides, right? We have something in IonQuant that combines multiple. We have this maxillary Q normalization actually in IonQuant. So the more peptides you have, the more robust quantification will be. MSTATs, of course, with multiple peptides will be able to eliminate outliers. So the challenge is always for single peptide identifications and quantification. And we think that through improved signal processing, we have better quantification. But importantly, we control the full. Again, I think the key advantage is that we are controlling match between run transfer FDR, right? So we are controlling transfer of those false IDs, and they are the problem. So if you're transferring a ton of quantification correctly, from a wrong peptide, and you're not able to eliminate this at the later stage, because all you get is just 1 or 2 peptides, you're going to get a lot of false quantification values, right? So that's why we're doing better. Now, manual quantification, well, sure, you can, if you're an expert person, you can look at the data and you can convince yourself that the tool is doing well. You can only do so much manually. So we use strategies such as using LFQ bench, using these organisms with multiple organisms mixed at known fractions, right? Or replicate runs, CV cross runs. You can utilize those strategies. But if you're an expert mass spectrometrist, you can of course convince yourself by looking. You can load in Skyline, in other ways, and see if we are quantifying it well or not. And in fact, we are working with many groups. We have a paper published in Molecular and Cellular Proteomics just a month ago in collaboration with Kerry Bacchus lab at UCLA, where they extensively used compared IonQuant against Skyline and benchmarked the tools against each other. They looked at they used Skyline a lot, and they benchmarked IonQuant, and both tools work well. And IonQuant was extensively benchmarked against Skyline. So I welcome you to look at this. Kerry Bacchus, Google me and find that it's a chemical proteomics application. MCP from a few months ago, a benchmarking against Skyline. So exactly like you were saying, a lot of manual processing, as well as a lot of manual analysis, a lot of benchmarks with, again, 4-to-1 labeling and 2-to-1 labeling, 1-to-1 labeling, and so on and so forth. Any other questions?
Yeah, just a logistics question. Several people asked if it's possible to have the slides after the presentation.
Yeah, Olga, just let me know. I think I shared just PDFs, right?
Just send this to me and I'll share. Thank you.
So I assume that the video is also going to be available too, right? Of that.
The video is available, yes.
Right. So, all right. So if no other questions to the TMT I track pipeline. So this is, we worked a lot on this because we are part of the CTTech consortium and we are really processing a ton of TMT data. So we worked on this for a while. And our pipeline has been used as part of 4 or 5 now landmark papers published through CTTech consortium. Some of these papers listed here, but of course also non-CTTech data sets as well. And again, this is where MS Fragger, Philosopher, now we use percolator more rather than peptide profit, but PTM profit for localization, protein profit, protein inference, quantification, extraction of quant, and then we have TMT integrator that's as part of FragPipe, but also MSStats is absolutely great tool for taking PSM TSV files from the pipeline and running through MSStats. So we have options. It's really nice to have great options for analyzing the data. So we have just to go through various steps, because every tool would go through that. We have TMT integrator that kind of pre-calculates some of these filters. So TMT integrator takes a PSM.tsv table from every data set, like MSStats would take PSM.tsv file from every plex, right? So each data set would have multiple plexes. If you have only one plex, that's of course, you just have one PSM table. If you have multiple TMT plexes, with or without bridging channel, then you'll have multiple PSM tables generated by FragPipe. And then you'll have to go through certain steps to filter the PSMs and then assemble them, group PSMs into proteins or peptides and aggregate the information and generate the reports, right? So, and the common steps are, reference normalization if we have a bridging channel, grouping, outlier removal, aggregation of PSMs, and then normalization. So, typically the PSM filters that we apply, and I think MSStats applying some of these filters, in the same way, but it also has more advanced modeling for some other things. So our basic PSM filters, our PSM tables we compute. Of course, we apply FDR-based filtering in the first place before we generate PSMs. PSM.tsv file is all quantification, but we calculate isotope purity score. We apply intensity cutoffs to eliminate PSMs with like less than 5, intensity that's less than the lowest 5% of the intensity in the data set, sort of we can eliminate by default. We use 2.5% for phosphoproteomics by default. The PSM tables have everything, but the filters that we apply in TMT integrator, that's where those filters are applied. We also select the best PSM among redundant PSMs per fraction in TMT integrator in whole proteome data. And then we group PSMs based on a specific index. And our index, we have multiple protein, peptide, G, site level index if it's a PTM data. And then we do outlier removal. If you have multiple PSMs for the same protein, we kind of see that the ratios, after we calculate the ratio to the reference channel, we aggregate PSMs to the protein, and you kind of try to establish sort of a median level across all PSMs, and in doing so, we eliminate outliers before we calculate the median. So, once we eliminate the outliers, we take the median of PSM level ratios to calculate the corresponding sort of ratio of the protein level. So it's a pretty simple strategy. MSStats would do a more elaborate modeling here, but TMT integrator by default does what I described here, basically aggregation of PSMs to a particular level, some normalization, outlier removal, followed by additional normalization after the fact, of which we have medium centering and more advanced, more aggressive normalization that we no longer recommend to do. Our median centering is sort of a standard normalization that we do. And you also have an interesting step that I don't have time to explain, but we have conversion of reference. Like, first we have a reference to a ratio to a reference sample, a bridging sample that we have. If you have a single plex, we use a virtual reference approach where kind of it's a reference to an average of all samples. But if you have multiple plexes and you need to merge them, the best way is to insert a reference sample that's a pool of all samples. Now, if you don't have that, you still can use a virtual reference approach. But anyways, everything is converted first for each PSM as a reference, as a ratio to that reference, whether it's a pooled sample or a virtual reference. So our final, our tables aggregated to the peptide or protein or gene level are first also ratios, right? It would be like for each protein, there would be a ratio to a reference. But to convert it back to the absolute abundance intensity, we have this one additional conversion step where we estimate abundance of peptides and proteins in the reference sample through several strategies. We use MS1 intensity for our default strategy. We calculate MS1 intensity of each peptide ion, apportion that intensity according to the PSM level ratios, reporter ion ratios, and calculate kind of like the MS1 intensity of the peptide and then the protein in a reference sample in each plex. And then we aggregate measurements, reference sample measurements across all plexes to get what we call reference intensity. So at the end, we calculate what we call abundance tables, where ratios to reference for each protein in each sample gets converted to abundance intensity, basically, in that sample. So we have a ratio table and abundance table. An abundance table is kind of like the absolute abundance in a way in each sample. So if having absolute abundance is useful for downstream statistical methods that would like to know what's the abundance of the protein is, not just a ratio, or if you want to correlate, say, against RNA-seq data for each sample within each sample, then you really, ratio is not going to do it. You have to have absolute abundance estimated, and for some other applications as well. So anyways, again, we, as usual, I'm running a little bit behind, so I'm going to probably cut the DIA data part a little bit, but of course we compared against a whole bunch of other tools, and you can find benchmarks published in literature as part of our papers, as well as we compare it in a collaboration with Binzang lab at Baylor, for example. For phosphodata, we have a paper on MCP a few months ago. We use some deep learning based benchmarks to evaluate TMT integrator with in FragPipe, or rather the whole pipeline, sort of not TMT integrator, the whole sort of our pipeline against a whole bunch of other tools, including MaxQuant. Here, this is our comparison for the CCRCC dataset from CPTAC, where you run through TMT integrator and MaxQuant would take you like 2 weeks to run it. TMT integrator will take you like 7 hours on a cohort like this. It's a huge cohort, 23 plexes, 24 fractions each, and still you can analyze it in half a day with our pipeline and 2 weeks with MaxQuant. But anyways, we have a much better correlation against RNA-seq data is measured, as an indirect measure of accuracy of quantification. Not to mention that, again, we quantify more proteins. We also, as part of CPTAC, we run this on 15 cohorts, so the pipeline scales to a ridiculous amount of data. So now it's like 15 CPTAC cohorts. It's just gazillion of data. And another advantage is that, again, we generate reports at various levels. Again, I don't have time to explain this now. Perhaps Philippe and Finchao can explain that later when they show the output. But we generate reports at gene, protein, peptide level, multi-site, single-site level. Single-site tables are if you do like a phospho analysis, and you want to only keep sites with confidently localized sites like PTM profit probability of 0.75 or above. So those will be only present in the single-site data. Multi-site data will contain unlocalized sites as well, and so on and so forth. So, and here is some information about how we generate those indices. In the case of phospho data, again, I don't think we have phospho really. I'm not going to explore as part of the sort of hands-on later that Felipe will do with Finchao. So, but all of this is described in our website. So we generate really, like I said, PTM level reports, where in a multi-site report, we keep all configurations localized, unlocalized, partially localized. But in a single site report, we'll only keep localized sites and only at a level, only like a one row, one site. Where in a multi-site report, for example, a peptide that has two phosphosites that are localized will be a different entry from the same peptide with only one of the two sites localized, or a monophosphorylated peptides. They will be all kept as separate entries in a site, in a multi-site report. And they will be collapsed to like a single entry in a single site report, one for each site, and only if it's localized. And this is a duplicate of the slide. You just saw that. We've done a lot of benchmarks in a non-CPTAC data, for example, looking at again in a human plus E. coli data set. It's an E. coli data set where there were human protein spike in at known ratios, right? So again, we compared against Protein Discover, MaxQuant with different settings, and to demonstrate, and we have MS2 data here and MS3 data. So this is something that will be, it's part of the manuscript that we are writing now, and that you should see soon. Hopefully it's a preprint as well, hopefully in the next month. But anyways, we show that both in MS3 data and MS2 data, we have really a good performance. Of course, we still have the ratio compression effects in MS3, like all tools have, but we also have various ways to minimize that. And we have comparison against Protein Discover, MaxQuant, to show that we quantify more proteins and the accuracy is comparable or better than other tools. So it's pretty much have 1 minute until the end of the hour. So I will just very briefly tell you that in addition to TMT, we fully comport DIA quantification. It's just, this is where we put a lot of efforts in the last few years. If you're familiar with our work in this area, that we sort of pioneered this direct DIA approach with DIA Empire, as well as the hybrid spectral library building approach, when the libraries are built from DIA data directly combined with DDA data, if we have that, to build a single library. So this is what we sort of published, a while back. People first did not believe in direct DIA, but now everyone thinks that's a really good strategy. So FragPipe supports fully DIA Empire-based workflow for building hybrid libraries. And now it's a one-stop workflow. We added Diane from Quantification in collaboration with Vadim Demichev. Diane is really a great tool. It's a part of FragPipe. So, it's just with a few clicks, you can load DIA data, you can load DDA data if you have. And then if you use DIA Empire workflow, it will run DIA Empire, extract through MS-Frag spectra, will go through MS-Frag research, MS Booster, DDA data will go through the same. Protein Prophet will all combine it into a single library with easy PQP, FDI filtered, and so on. And then we'll go to DIA data with Diane and extract quantification. So one-stop analysis. But before you get all too excited about DIA Empire, we have a new tool called MS-Fragger DIA that now MS-Fragger can do direct DIA analysis directly, and the results are just outstanding. So again, all benchmarks, not only we of course beat our own tool DIA Empire strategy, but we also perform really well against all other tools, including Spectronaut. And again, you can do it with just a few clicks, load DDA, load DIA. MS-Fragger will recognize what's DIA. Well, you have to annotate them to DIA or DDA, but MS-Fragger will automatically run DIA data through MS-Fragger DIA tool. The DDA will go through the conventional DDA algorithm, deep learning rescoring with deep learning with MS Booster, percolator rescoring, peptide profit, easy PQP for spectral library building. And then given that combined hybrid spectral library, again, Diane will take the DIA data and will match against that library and will extract quant. So this is how it will look. You'll see that in FragPipe 17, it's already available. I have a DIA Amparo already separate. Sorry, and Diane is a separate tab here, DIA Amparo as well. But all you need to do is to load like DIA spec deep quant workflow, load DIA and DDA data, annotate DIA runs as DIA, DDA is DDA, and then click run, and the whole thing will run all the way to through Diane. Now, if you only want to use, for example, some runs for DIA for quantification and not for building spectral library, you can annotate them as DIA quant. In that case, the DIA runs will not be used for spectral library building. If it's as an option. If you do GFP, this is actually great. The stuff that Mike Macas, developed, really great ways to generate DIA data with a gas phase fractionation strategy for building the libraries. So you can load gas phase fractionated data, set it as to GFP DIA type, and load wide window DIA data, annotate as DIA, and then build a library from all data combined. It works great as well. So I had some benchmarks. Let me skip that. Just say that, again, this is a particular phosphor, very deep phosphor data set that we, compare to Diane, library free. So Diane also, and Diane is a really great tool. All tools here are great. Spectronaut is also good. And Diane is good, all have advantages, but of course, we have to show that we do better than other tools, right? Otherwise, why would I show this? So don't trust me, go and do it yourself. Don't trust any bioinformatics group, whatever they show you, run it, compare yourself, and then decide for yourself. But in our comparisons, we do really well. So here is this number of phosphosites quantified compared to library-free. If you run a library-free Diane followed by quantification versus MS-Frager-based library building followed by Diane quantification. So this is MS-Frager DIA plus Diane. So we can quantify more peptides, certainly more than Spectronaut, and outperform DIA Amparo as well. But DIA Amparo has still advantages for certain applications. Don't have time to discuss this, but there is a lot of advantages for DIA analysis with DIA Amparo as well. For like non-specific searches, for example, still DIA Amparo based. I recommend it. Now, for long UI input single cell data, we are now writing a paper on MS-Frager DIA. So, again, we show against Spectronaut, for example, on 5 cells, 10 cells, 50 cells. We also have single cell data where we show that through our MS-Frager DIA strategy, we significantly outperform Spectronaut, not only as far as the number of IDs, but we also get better quantification. And that's because Diane is absolutely outstanding tool for quantification. So it's that. So here I'm showing 5 10 cell data from a recently published data from our collaborators from UG Chen lab. I just spent a month in Taiwan again, met with them, a really great group. So we are also working with them, adapting our tools to the single cell low input data. And they have a really nice technology of single cell analysis, DIA based, that we are collaborating on and running our tools on the data. It works well. If you're on Twitter, and you should go and follow me on Twitter, because I advertise on Twitter like there is no tomorrow, because I'm, where else is Twitter for, right? Other than just shameless self-promotion, right? So here is, I occasionally download the data sets and run through the pipeline and see the results. So I got excited about this ultra deep DIA publication from a few days ago. Here is the reference in Journal of Proteome Research. So I downloaded, run through MS-Frager DIA, 3 replicates of 3 replicates of cell line, 120-minute gradient, 3 replicates, and 10,000 proteins quantified, over 8,500 quantified with CV less than 10% with MS-Frager DIA coupled with Diane. So works well. Team staff data, maybe you've seen a paper from Matthias Mann lab on single cell proteomics. So they adopted FragPipe and Diane. So basically they adopted our pipeline, which is, I don't need to go and show you our results. If if Matthias Mann lab thinks that our pipeline works well, it probably works well. But again, don't trust what I'm saying. Take your data, run it, and compare with other tools, because other tools are also good at, and see what works best for you. So, interactome analysis, the output from our pipeline fully compatible with our reprint resource. So you can load everything. If you do interactome studies, take the output from FragPipe, quantify it using spectral counts or ion quant intensities, load and reprint reprint the PMS for interactome analysis and scoring, as well as full support for chemical proteomics workflow, which is something we're very excited, but it's way too complicated to explain, especially because I'm out of time. So, again, thank you for listening for so long for me. I talked about FragPipe from our workflows for post-translational modification analysis to quantification that we do, whether it's label-free quantification or TMT or DIA, but we also support SciLac and advantages of the pipeline when it comes to speed and accuracy, and all can be downloaded from this link. FragPipe 18 coming out soon. And again, sincere apologies. If you know me, I'm always run out of time. This is my problem. I should do something about it. Well, maybe next time. There's always next time, right? So I'll stop here, and if you have any time for questions, I'll be happy to answer them.
Well, thank you so much, Alexei. I know you have to leave soon. Can you tell us how much time you have?
No, I have time. It's how much time you have. I'm okay.
Okay. So, well, there's a couple of questions. So, like, I don't even know where to start. So maybe talk about the PTM identification with MS Fragger, FIA, and Diane. And for example, deamidation. If you can comment.
Deamidation. So with what quantification workflow? So, we support DIA, TMT, label-free, all of that, right? If the MS Fragger is just a search engine that will identify them, identify the phosphopeptides. Now, then how you could generate this? These are sort of the reports, right? If it's label-free, we have site level reports that are generated by ion quant. So you'll, maybe Finchao can show you that. And ion quant will take, if you run PTM Profit, ion quant will look at the localization probability of 0.75 for deciding which sites are localized, and will generate the reports. If it's a TMT workflow, if it's PTM Profit that's used for localization, TMT integrator will use that, and you'll have these reports. If it's SciLac, we have SciLac as well. Ion quant does SciLac and regenerates the reports. And if it's DIA, that's a little bit of a partial solution right now. We'll build the libraries that contain the phosphosites, and Diane will, then Diane has ways to quantify the phosphopeptides and report some additional information that's useful for knowing, how confidently it was with extracting fragments specifically for those phosphosites. DIA with PTM analysis is still quite challenging, actually, whatever the pipeline you use. So it's a work in progress, I would say, but certainly Diane is doing a pretty good job already. And our part on spectral library building part will build the libraries for phosphopeptides.
And is there a limit on the number of PTM for a search? I guess it relates to DIA.
Well, for the convention, if you do the regular DDA search, DDA data, or DDA or DIA, a closed search, right? We have 7 or 9 variable modifications or something that we allow by default. More than that, but PTM analysis is our bread and butter. So we work with groups to do all sort of crazy stuff. That's why we have common mass offset workflows. We have labile workflows. we can search. Maybe if Chao can, you can show and I can stay a little bit maybe in that comments too. If you open the workflow, if you open the FragPipe, you can open a mass offset workflow. You can put common mass offsets, like you can put multiple mass offsets there. Even with phospho, you often do it through the mass offset strategy because phospho is a low amplification. So FragPipe is super powerful for that because there is a lot of advanced options. So it supports all the basic stuff that you know from any other search engine. Put 7 or 9 variable amplifications and do it that way, right? But if it's more than that, I suggest you start learning mass offset workflows, how to specify mass offsets and go through that, because then there is no limit, right? Then or you can do the open search, then you can search through everything in one go.
And there's a question that came up when you talked about TMT. So could you comment on the number of identifications between MS2 and MS3?
Okay, well, the number of identifications always drops if you do MS3 based. I mean, I want to say probably the number of PSMs maybe will drop, 20-30% at least. The number of proteins that drop will be less, maybe 15-20%. Yeah, there is certainly a drop in the number of IDs if you have to acquire additional MS3 data, right? That's why CPTech, for example, uses MS2 only approach. Now, the price you pay is that, of course, there's a much bigger ratio suppression effect. So with MS2-based TMT analysis, if it's a tenfold change, biological change, you'll be likely to get 2-3-fold change from TMT analysis, right? MS3 would allow you to get a more realistic fold change, a more accurate fold change. But the price you pay is the loss the number of IDs. I'm not a big believer in real-time search engine. I should rephrase it. I mean, I think we need to explore the role of real-time search as part of MS3 workflows. I don't believe in it. No, I'm sorry for saying that. I think it has limitations, but it has advantages too, right? As a way to boost the MS3 workflow. So there are pluses and minuses. So I think you need to decide what's more accurate, what's more important for you, the accuracy of quantification or the number of IDs. In our facility, for example, we mostly do MS3 based because we don't care how many proteins you quantify. We want to have more accurate and more reflective fold change, reflective of the biology. But a lot of times, you just want to differentiate between changing and not changing, and MS3 workflow is just fine, and you get more IDs.
Thanks so much. So we actually have a few more minutes. So for the participants, this is your chance to ask questions to Alexei, or if you maybe would like to hear a little bit more details on some of the previous slides, that is the moment. So let's see if you have. So I think we have a few more minutes on the schedule. So let's give it a shot. And maybe while we're waiting for more questions, Feng Chao and Felipe, could you maybe say a few words about what is planned for the hands-on more specifically? So what type of workflows also, so that everybody can have a preview?
Yeah, absolutely. So we're going to show how to, well, we're going to show a few things. First, we're going to demonstrate how to find our tools and our programs on the internet, how to work with the websites, how to download them. Then we're going to show how to configure a RAM using a simple DDA TMT MS3 experiment, how to operate the program, how to put all the parameters in place, and then we're going to go through the output files and explain how you can use them.
And I don't think we have anything on PTM. Do we have anything on PTM? Well, I guess this is on quantification, right? Focused. So so that's what you have. Mm-hmm. Well, it's the good thing about FragPipe is that it's very intuitive. So everyone who would like to use it for a particular workflow, and again, our FragPipe is a knife sort of proteomics workflows. because we support pretty much everything. We don't have cross-linking now. Maybe we should, at some point add that. But you just select the workflow that most similar to what you want to do, or exactly what you want to do, load it, and the things already set for you there. And TMT is just one of the workflows, or rather we have multiple workflows, because TMT 10, 16, TMT phospho, TMT ubiquitination, and so on.
So going back to MS2 versus MS3, can you comment on why MS3 spectra are better for quantification?
Well, yes, because so how we quantify in TMT is that we have this reporter ions in the mass spectrum that basically, in principle, reflect abundance of reporter ions, for a particular sample. That's what it should be measuring, the abundance of that peptide in that sample. But the problem is that we are not isolating just one peptide for MSMS. We are isolating a whole bunch of ions with a window, of say ±1 delta, right? You can narrow it down with Eclipse Explorers. You can successfully or Lumos narrow down to 0.8 delta, and with all the fusions you have to go with higher windows because the ion optics wasn't that great. So, but there's still a pretty wide window. So you're actually isolating not just the target peptide, but possibly some co-fragmenting peptides. So you're identifying the peptide on which you acquire the SMS, but the reporter ion intensities reflect the presence of those co-isolated ions, other peptides, and their abundance, right? So they also contribute to those reporter ions. So and that's why you have the ratio suppression effect and some inaccurate quantification in MS2 based TMT data. In MS3, you take one additional step of actually quantifying from MS3, where you will select Y and B ions, or expected Y and B ions, from the peptide on which you're acquiring the spectrum, really, right? And collect MS spectrum with MS3 spectrum on that peptide, and the reporter ions in MS3 then will be really reflecting just the peptide that you identified. I mean, that's also not perfect. There is also some interference, but by this additional isolation, sort of the isolation step, you're removing some of these interferences from quantification.
Sounds great. And kind of along similar lines, how reliable is DIA for identification of PTMs, in particular for weak ions in the phosphopeptides and mixed spectras, given that we have a wider isolation window?
Yeah, so, I mean, we get a really good number of IDs. How reliable, if the question is the number of, I think the accuracy of quantification, that's where it's a really good question. So I don't have a really good answer right now. I have not seen a lot of good benchmarking data sets to really look at the accuracy of quantification of, say, for phosphodata, DIA versus DTA. I've seen the number of IDs in direct analysis. If you look at the signaling pathways from, say, maybe differentially expressed phosphosites in DIA versus DDA, those would show a comparable picture, and the number of missing values would be less by the nature of your sort of more targeted extraction of quantification. But I don't know. I think it's a work in progress. I think more needs to be done to see how accurate we can be with DIA, specifically for PTM containing peptides.
So another question, Silas is asking, for curiosity's sake, how much of the data from a typical proteomics run, for example, DDA on Orbitrap, is explained by standard search settings? Or how common, abundant, and common is it to find PTMs in typical standard proteomic data? Yeah, so a really good question. This again goes back to where I started in my career in this field of, what are those unassigned high quality spectra? And that's,
again, going back 20 years ago, you know, LTQs, not LTQs, LCQs, like 2000, 2001, right? You would probably get only 15% of MSMS scans identified with high confidence, right? 10, 15%, if not less, in some challenging data sets. But a lot of that was a lot of not identified things was just noise. You know, these days, I would say probably we're getting closer to 30, 40% of spectra that can be identified in a regular search with only looking for, well, maybe 30% looking for common modifications like oxidation and so on. If we do open search, we'll boost the numbers, right? Because we'll get all these chemical artifacts, as well as capture non-specific, you know, the neutral losses, some tryptic peptides, and so on and so forth. And probably will get to maybe 45%, 50%. You know, still a large portion of MSMS spectra not matched. Now, out of that half that's still not matched, there is a lot of low quality data because the instruments are fast. So, you know, you're often just acquiring MSMS on, you know, on just nothing, right? Because there was no a good strong signal sort of to acquire MSMS. But there is also a lot of high quality spectra still left. So even with open searching, with everything, you know, we go to, okay, so maybe you explain 60% of all spectra, you know, but probably explaining 70, 80% of high quality spectra. So what are those extra 20%? Yeah, you know, they are mutations. So, you know, open search will find them with some mesh shifts that, you know, we may not be very confident about them on open search. And that's why, you know, they're not passing FDR because we're pretty stringent, you know, but, you know, we've done, it's a big progress in how much we can identify now, especially with open common method search strategies. But still, there is more to identify with improved scoring, with improved sequence database generation to capture those variant peptides and novel peptides. I think there is still room to gain, room to improve.
So somewhat related. Now, when things get more difficult, in particular in single cell workflows, have you tried identifying PTM?
Yeah, of course. I mean, the difficulties are sort of, we're not really interested in, you know, if for simple stuff, you can, you know, just go and run your favorite protein discovery tool if you have it. Actually, I should take it back. Now our pipeline is so good that, you know, you should run it for everything, not only simple stuff. But what we're really interested in is the difficult stuff. So, you know, when we get the data like single cell, the first thing, you know, I usually try to do is to run open search. And it's, you know, we find a lot of interesting stuff even in single cell data. So some of that, of course, you find post-translational modifications too. You do see phosphorylation and in ubiquitination, even in single cell data. Not a whole lot, because of course you're not starting with a lot of stuff to begin with. But we see some interesting artifacts because single cell workflows like nanopods or some, you know, they require different type of materials, different tubing, different. So we see some polymer contamination, for example, that you would not see in a traditional workflow necessarily. So we see some interesting mesh shifts. So I gave a talk in one of these. I think I gave a talk on ASMS single cell workshop, and I think I'll speak again in Boston actually in a month on single cell. I may show some slides of things that we find in single cell data, and some of them artifacts that are just related to the type of tubing, you know, that you use in single cell workflows to minimize the sample losses, or some type of reagents that people use. And I talked to Ryan Kelly about this in Inju and Thermo people. So they all like scratch their head. Oh, okay, that's where it's coming from. Okay, so yeah, there is a lot of interesting stuff. So if you get our pipelines, you can see all of that for yourself.
Well, and one more question, going back to TMT. So there was a paper where authors showed that if you take TMT balancer I and conjugate it to peptide for quantification, it is better than MS3. Mm-hmm. Do you happen to have an opinion about that?
You know, yeah. So I talked to What? I
So
talked
there's
to the
a-
authors of the.
There is a link to this paper. I can put it in the chat.
I'm just. I know who you're talking about. I'm just. I need to go back to. I'm going to stop sharing so I could see it in the chat.
Yeah.
Right, Martin. Yeah, I talked to him at his poster a few years ago. I had a really good discussion with him. I think these guys are doing really interesting work. So we haven't looked at that much because my understanding is that the commercial versions of the instruments somehow are not supporting this type of workflow really well yet. Well, anyways, I think this is exciting. This is interesting. If people start using it or if the data is available, we'll be the first one to look at that, or not the first ones, but we'll be super interested in looking on that. So if someone has good, interesting data already, if it works, if you can generate this data, send us email. We would like to take a look at the data and see what we need to do to support it.
Okay, so there's a couple of more technical questions, but I think we can answer them just in writing. So I think this is a good time for us to take a bit of a break. So I would suggest that our presenters, if you see questions which you can answer in the Q&A in writing, please do so. And otherwise, we will take a 15-minute break and we will be back at 12:45 Eastern, so quarter before the hour, wherever your time zone is. And then we will continue with the hands-on by Felipe and Feng Chao. And Alexei, if you can stay around.
I will log out, but then I'll try to come back. And thank you all again for organizing such a terrific course. I mean, I'm glad to see so many people participating. And thank you all for your interest. And I hope that some of you will be users of new users of our pipeline.
And maybe I can volunteer if you come to